So I'm Sherri Jones. I'm a postdoctoral fellow in the lab of Thuong V Nguyen at the MUHC. I'm currently doing human research, but I have extensive experience in animal models, so manipulating hormone levels prenatally and postnatally, and looking at the effects of hormone manipulations on sexually dimorphic behavior. And so today I'm going to talk to you about the 4 core genotypes. So first we're going to talk about what actually causes sex differences in health and disease, and to do that I'm going to give you a little review of the development of sex differences, and it's important to understand how normal sex differences develop to understand the 4 core genotypes. So I'll talk about how we can actually disentangle chromosomal effects from hormone effects using the 4 core genotypes model, and then how we can address that in this very simple 2x2 design. And then if there's time, I'll go through one neuroscience example, and then I'll tell you how you can actually do it. So first of all, which of these two is a chromosomal male? Who's pointing this way? Anybody pointing this way? Oh, that's good. Okay, good. So, right. So you're right. So it's the XY, and I'm glad that everybody's got that basic piece of knowledge. So what causes sex differences in health and disease? If I were to ask you, can you name a couple things? What causes sex differences? Hormones? Good. Is there anything else? Sorry, chromosomes, did you say? Good, this is great. Okay, so right, so in mammals, sex differences are due to sex chromosomes, which are sex determining, and or gonadal hormones, which are sexually differentiating. So here's our Y chromosome, and on the Y chromosome, right, which is in males, we have this SRY gene, which is called the sex determining region of Y. And what the SRY gene does is it codes for a protein called testes determining factor, which leads to the undifferentiated gonads to develop into testes. So we all looked like this back in utero, okay? We both had these undifferentiated gonads. We had the Wolffian ducts and the Mullerian ducts. And in males, the SRY gene promoted the differentiation of the undifferentiated gonad into a testes, okay? And then and so TDF, the testes determining factor, promotes the undifferentiated gonad to differentiate into testes. And one of the earliest steps in testes development is steroidogenesis, so the production of steroid hormones. And so testosterone, which is an androgen, is upregulated. And what that does, and there's also an upregulation of anti-Mullerian hormone. And what that does is that causes the Wolffian ducts to be maintained, and it causes the degeneration, so anti-Mullerian, so the Mullerian ducts are like the female system, the anti-Mullerian hormone causes the degradation of the Mullerian ducts. And so that's how you get differentiation. And so in the normal female, in the absence of the SRY gene and the testes determining factor, the Wolffian ducts, so the male duct system, degenerates, and the Mullerian ducts survives and differentiates. Okay, so what that means then is it's this SRY gene, a functional SRY gene and testes determining factor, that leads to biological sex differences in the gonadal systems. Okay, so is that pretty clear? Everyone agrees with this? Good. Didn't make any mistakes? That's good. Okay, so in the absence of, as I just mentioned, in the absence of a functional SRY gene or a functional TDF, the female system develops. So this has classically been referred to as the default system, right? Females are the default system. So I'm not going to argue too much with that right now. Just suffice it to say that it's actually not quite true that it's default. There's an active process that occurs, but I'm not going to talk about that today. So basically, somatic sex, so because in the absence of a functional SRY gene, you can actually get, from a Y chromosome, you can actually get the development of a normal female gonadal system. Somatic sex is secondary to and dissociable from genetic sex. Okay, so we're going to come back to this later, just keep that in mind. Does that make sense to everybody? Okay. So there's 2 classes of sex-specific signals. There's the signals that occur during the organizational period, so the perinatal period, under the influence of perinatal testosterone from the male testes. And then there's the activational period, which occurs under the influence of gonadal hormones at puberty. So basically what that means, up here I'm showing you a slide of the human organizational effects of sex steroid hormones. So here on the y-axis you have the level of, so the dotted line is testosterone levels, and this solid line is just the brain sensitivity to the steroid organizational effects of the steroid hormones. So the brain is very sensitive to the organizational effects of testosterone early in life, and then that teeters off and into adulthood. So this is basically the critical period is what I'm trying to say. And then this is in the rodent model. It's essentially the same figure showing that there's this sensitive period where testosterone organizes the brain into a male brain, and that organization that's set down prenatally is then activated in adulthood with the activation of puberty. Does this make sense to everybody? So there's these two distinct periods, one which is organizational during the perinatal period, which is thought to promote a permanent organization of the brain and subsequently sexually differentiated behaviors. And then there's the activation of those systems that were organized perinatally that become activated under the influence of gonadal hormones, right? So actually rodents are really model systems to study this because their sexual behavior is completely dependent on hormones. So in female rats, they will show, or mice or other rodents, they will show this like classic lordosis posture only around the time of ovulation when estradiol and progesterone are high. And the rest of their estrous cycle, they will not show these behaviors. Similarly, if you take out their gonads, so if you overrectomize them, they will not show the sexual behavior, but you can reinstate it by giving them exogenous administration of estradiol and progesterone. Same thing for the male. So this sexual behavior is dependent on testosterone because if you remove the testosterone, they no longer show the behavior, and then you can reinstate it by readministering testosterone. Does that make sense? Everybody's following? Okay. So there are basically 3 independent processes of sexual differentiation. So remember, the sexual differentiation happens in the perinatal period under the influence of testosterone. So the 3 processes are feminization, masculinization, and defeminization. In females, you have only feminization that occurs because this is that default system, right? So feminization basically occurs in the absence of SRY and in the absence of testicular androgens, and it creates neural processes that support female typical behavioral patterns and reproductive physiology. In males, you have these 2 processes that occur, defeminization and masculinization. So masculinization is induced by testicular androgens, so the testosterone released from the fetal testes, and it creates neural processes that support male typical behavioral patterns and reproductive physiology. And then there's this defeminization that also occurs, and it's either the removal or the suppression of neural underpinnings that promote female sexual behavioral patterns and physiology. So basically you have to turn off the female default. Okay, that's what this is. Whether it's actually a removal or a suppression, we're not actually clear. There's some debate in the literature. Some of my data suggests that it's actually a suppression, but I'm not going to get into that. But this is just to kind of reiterate that female system is the default process, and in males you have the process of masculinization and defeminization. And so you can manipulate hormone environments and then look at these behavioral patterns to look at how the hormones affect these sexually differentiated behaviors. So classically, people have studied sex differences on the hormone side of things. So you can look either at the organizational period, so in that early life period, by, for example, treating neonatal females with testosterone, which would masculinize the females and defeminize them. Or you can deprive neonatal males of normal testicular secretions by gonadectomizing them early in life, or administering, for example, androgen receptor blockers so that testosterone cannot have its effect. Or you can look more in adulthood, so to block the activational effects or manipulating the activational effects. So you can, for example, manipulate them by gonadectomizing the animals and then and or replacing the hormones, so giving hormone replacement. So this is classically how it's done. The problem is, not all sex differences are explained by hormones. So how can we actually disentangle chromosome effects from hormone effects? And this is where the 4-core genotypes model comes in. Okay, so this model was developed by Paul Bourgogne and Robin Lovell-Badge at the Francis Crick Institute. And there's basically two critical genetic components that have to happen in this model for the four-core genotypes model to work in the four-core genotypes model. So the first thing is the deletion of the testes-determining gene SRY from the Y chromosome. And then the second is the insertion of a functional SRY gene onto an autosome in the same mouse. Okay, so that's shown here. So remember, our male mice have an X and a Y chromosome. So what you do is you take the SRY gene out, which remember codes for the testes determining factor. You take it out of the Y chromosome and you move it into an autosome. And so typically male mice make two types of sperm, an X sperm or a Y sperm. But these four-core genotype mice make four kinds of sperm. So you can have, this is like your typical female or female sperm, I guess X sperm. So this is just the X chromosome with the chromosome 3, typical. If you were to mate that with a female mouse, you would get offspring with ovaries. There's a type of sperm that would have the X chromosome and the SRY gene implanted on the autosome, like chromosome 3. Because this is an X chromosome, but there's an SRY gene on one of the autosomes, the SRY gene, functional SRY gene, would then lead to an offspring. If this male mated and this sperm fertilized, it would lead to an offspring with testicles. So it would have a X chromosome, so XX chromosome, but testicles. Does that make sense? Or you can have a sperm with that Y chromosome deleted, and chromosome 3 with no SRY gene, which, as I said, would default, because there's no SRY, would default into a female phenotype, so there would be ovaries. Or you can have the Y chromosome with no SRY. So sorry, we represent that by Y minus, meaning it's the Y minus the SRY gene. And with the SRY gene inserted on chromosome 3. So this is kind of like a male, if you will. Okay, because the SRY gene is functional on chromosome 3, and so testes develop. Does that make sense? So these are the four types of sperm you get. And so if you were to then mate that father with an XX mother, so mothers are always XX, if you were to think then that, so these are the four possible offspring. So you can think of this as the first X chromosome coming from the mother, and the second sex chromosome coming from the father, where it's either an XX, so it's like a typical female with ovaries, and we refer to that as an XX female. An XY minus, which is a male or a Y chromosome that's lacking the SRY gene. Because it's lacking, the genome is XY, but the gonads differentiate into ovaries. And we refer to this as an XY female. So the sex here that we're referring to refers to the gonads, okay? So this would be XY female. Or you can have an XX with an SRY inserted on the autosome, which gives you a testes. So we refer to this as an XX male. Or you can have the XY minus with the SRY inserted on the autosome, which would then give you a male, so XY male. So what we would classically, kind of like a typical male. Does that make sense? So this is where the four-core genotypes come in. So this is just another representation of that. So again, you cross the XX mother with the four-core genotype father. You get these four different genotypes. We abbreviate them as XXF, XYF, XXM, XYM. The gonads of the animals, two of them will have ovaries. So those that are lacking the SRY gene develop ovaries. Those that have the SRY gene on the autosome develop testes. Okay? And then what we can do is a simple 2x2 design. So simple, eh? So simple. I know nothing of genetics. I could not do this, but it's so simple. Okay? So basically now you've got two mice that have different sex chromosomes, so female sex chromosome, male sex chromosomes, but they both have female gonads. And you have two mice that have different female, male chromosomes, but have male gonads. And then you can do a 2x2 where you compare the XX animals versus the XY animals. And if there's a difference, then you have a chromosome effect. And you can compare mice with the different gonads, so the female gonads versus the male gonads. And if you have an effect, then you say it's a gonad effect or sex effect. This is the same thing represented in different colors because sometimes we just want to see it a few different times. Okay, so it's exactly the same thing I just said. Does this make sense to everybody? It's genius, isn't it? Okay, it's very cool. Okay, we can only do this in mice, by the way, not rats, yet at least, so it's a mouse model thing. The other cool thing about this model is when we have an ANOVA and we do a 2x2, what's the other cool thing about ANOVAs with 2x2 designs? We can look at interactions. Huh? Okay, so this is fun. So this is just an example of how you can actually use the 4-core genotypes model to look at the interaction effects, so the combined effect of gonadal hormones and sex hormones. And this is a study that was published in 2006 by Gatewood et al. In Journal of Neuroscience. And this is basically using the 4-core genotypes model in an aggression study. So this is the latency to attack an intruder across a number of trials. And each of the 4 categories here are the 4 genotypes, 4-core genotypes. And down here on this bottom panel, it's the percent of attacking, so the percent in each group attacking on the first trial, again, by the 4-core genotypes. And an interaction was detected where it's only animals with an XX chromosome and female gonads, so ovaries, that displayed a longer latency to attack, and fewer of those animals attacked on the first trial. So this is an example of how you can actually use the 4-core genotypes to investigate sex differences further and look at the combined contribution of sex and chromosome, or gonads and chromosome. Okay? So this is just a summary, just repeating again what I've said a few times. The 4-core genotypes model, what's cool about it is that it produces X, Y, and XX gonadal males, XX and XY gonadal females. And so then it allows you then to test the effects of XX and XY genotypes in mice that have the same gonads. Okay? It also allows you to look at the gonadal effects or the hormonal effects. So I mean, I didn't talk too much about it, but you can. There's 2 different ways of looking at of using this 4-core genotype. One way is to just use the animals as I've been explaining them, but another way is to actually gonadectomize the animals, and then you're simply looking at the effects of the gonads, the presence or absence of gonads, so you don't have that additional confound of the hormones released because of the feedback systems. Okay, and then it also allows you to look at the interaction of sex chromosomes and gonadal hormones. Did you learn something? It's good, eh? It's super cool. Okay, so if we have time, do we have time to go through an example? Where is Felicia? Yeah, okay, thanks. Okay, so just a really, really quick example in neuroscience. So this is a paper, some of you might know this group. So it's a group in Toronto where they looked at the separate effects of sex hormones and sex chromosomes on brain structure and function using high-field MRI in mice, so in the 4-core genotypes model, and then looked also at spatial navigation. So they basically used high-field MRI and did a voxel-wise whole brain analysis of brain structures in the 4-core genotypes mice. So this is a mouse brain, and these are different slices throughout the mouse brain, so A, B, C, D. So we're going from like the cerebellum to the forebrain, and so that's just represented here. So you've got like, you know, A corresponds to A, B corresponds to B, so as you're going through the brain. This top panel up here is showing the effects of sex steroids on different brain structure volumes. Here it's showing the effects of chromosomes on the brain structure volumes. Here it's showing the combined effect of the sex steroids and the chromosomes, controlling for training in the spatial navigation task. And then this bottom panel is just like your atlas showing what the different brain structures are. And so what they found is actually really interesting because they found, and so they looked in like 30 sexually dimorphic brain regions, and they found that the areas that are influenced by sex steroids are essentially independent from those that are influenced by sex chromosomes. That's amazing, okay? So what they found, they found regions like the periaqueductal gray up here, the parietal lobe, and the amygdala, and the amygdala is over here, and the hypothalamus is over here. These are areas that were influenced by sex steroid hormones. So blue means that females had a large, so the female, the animals with the female gonads had a larger brain structure than the males, and the more warm colors, so like the reds, means that the males had a larger brain structure than the females. And so those are the areas that they found were influenced by the presence of the gonads. Those that were influenced by the chromosomes was the occipital lobe, the corpus callosum, so this little thing over here, and the basal forebrain, so over here. So again, the blue colors, the colder colors, are larger in XX animals than the XY animals, and the warmer colors are larger in the XY animals compared to the XX animals. This is just looking at the combined effect of sex steroids and chromosomes, and they found that there were additive effects of the sex steroids and the chromosomes in the cerebellar cortex and in the superior colliculus. So this is I think this is a pretty amazing study because it shows, well, first of all, it shows the advantage of using this 4-core genotype model and how it can help us to better understand the relative contributions of gonadal hormones and chromosomes on sex differences in the brain. And then when they looked at spatial learning and memory task, so this is on the y-axis, we have the number of correct visits in a radial arm maze across trials. What they found, and here this is the effect of sex, or the effect of the presence of gonads, and the effect of chromosomes. So they found that all the animals improved across tasks, and this is kind of typical, but males, those with gonadal males, I should say, so males that had testicles, they learned faster than animals that had ovaries, okay? And there was no effect of chromosome. So this is an example, too, of how the 4-core genotypes can actually reveal some sex differences that we may not have otherwise found. So anybody wants to go now and do the 4-core genotypes model? Okay, I don't know if you do or not, but if you do, your question would probably be, well, how can I do it? So I would basically just refer you, if you're more interested in the 4-core genotypes model. Art Arnold is in the US at UCLA, and he's probably one of the largest proponents of the 4-core genotype model here in America. And you can go to his website to get more information about the model, but also on how you can order animals. And he's also very open to talking about this, so feel free to email him. Drop my name if you want to. Or you can order them from the Jackson Lab or UC Davis, and those are the websites. While I'm here, I also just want to mention, so Crystal mentioned before that we have the IGH trainee network. I'm the trainee lead at McGill, and so it's good to see you, Danielle. So I encourage you all to follow us on Twitter and Facebook. So basically the IGH trainee network promotes sex and gender research. I'm kind of your go-to person if you want at McGill. But don't be afraid to actually join the trainee network yourself. All you need to do is email Cora. Read about the responsibilities, but then join the trainee network. It's a lot of fun. It networks people all together so that we can talk more about sex and gender. And actually, it's really nice to see that we have some of our Université de Montréal colleagues here, so Danielle and Dr. Robert Paul Jester. We're actually trying to organize an event for the winter, so keep your eyes peeled for that. It would be great to have all of you there, and just kind of a fun way of sharing our research. And with that, just say thank you, and I'm open to taking any questions.